for today. Everyone, our demo for today is about acid-fast staining Kinyoun method or cold method. Before entering the laboratory, it is necessary to wear the PPE to protect ourselves from the chemicals or substances that could harm us. Here, I'm wearing my gloves, mask, hairnet, and oh, I'm sorry, I don't have protective goggles for now, but remember, in actual laboratory, it is necessary to wear it. Before we proceed to the staining procedure, we should observe the proper aseptic technique. Disinfect the table first, and wipe it. Okay, so let's put some papers on the table just in case we spill something. Before we start the demo, let me just give you some information about what we are going to do. We are going to do the acid-fast staining in Kinyoun method that is also called cold method. Kinyoun staining is developed by Joseph J. Kinyoun. In acid-fast staining, we stain bacteria that have mycolic acid. Mycolic acid are long fatty acids found in cell walls of Mycolata taxon, a group of bacteria that includes Mycobacterium. In Kinyoun acid-fast staining, we don't use heat, unlike in Zell-Nielsen staining that uses heat. Now, we are ready to start the Kinyoun acid-fast staining. Are you ready? So the materials we need are prepared bacterial smear, Kinyoun carbofixin, decolorizer, and in this demo, we are going to use TB decolorizer which is based on our reference, and methylene blue as counter stain and distilled water. Now, let's start! The first procedure is prepare a bacterial smear, but we're not gonna do that. Instead, let's imagine that I already prepared the fixed bacterial smear, and our specimen for this demo is Mycobacterium. The second procedure is flood the smear with Kinyoun carbofixin stain for 5 minutes. Kinyoun carbofixin stain is used as primary stain dye to detect the acid-fast bacteria because it is more soluble in cell wall lipids than in acid alcohol.
5 minutes later.
After 5 minutes, rinse it with deionized or distilled water. But in this demo, I will be using distilled water. After rinsing with distilled water, now I'm going to decolorize it using TB decolorizer, based on our reference for 3 minutes. In some reference, they use acid alcohol as decolorizer.
A few minutes later.
After 3 minutes, we will rinse it again using distilled water. After that, repeat the decolorization for 1 to 2 minutes or until no red stain appears. So let's say that there are still some retaining red stain, therefore I'm going to decolorize it again.
A few minutes later.
I'm going to rinse it again with distilled water. After rinsing, we will flood the smear with methylene blue as the counter stain for 1 to 3 minutes.
A few minutes later.
After that, for the last time, we will rinse it again with distilled water and allow it to air dry. Do not blot. After that, we are done. We can now examine the mycobacterium using low power objective and confirm at 100 times under oil immersion for presence of acid-fast bacilli. Expected result is that acid-fast positive organisms stain pink to red, mycobacterium will appear red, small, and lightly curved. Non-acid-fast organism, such as other bacteria and cells, will stain blue with methylene blue counter stain. That's all. I hope that you learned something. Thank you.